Welcome, and thank you for standing by. At this time, all participants are in a listen-only mode for the duration of today's conference. Today's conference is being recorded. If you have any objections, you may disconnect at this time. And now I'd like to turn today's meeting over to Dr. Ben Beard, CDC. Thank you. You may begin.
Thank you. Good morning or good afternoon, depending on where you're calling in from. My name is Dr. Ben Beard. I'm Chief of the Bacterial Diseases Branch in CDC's Division of Vector-Borne Diseases. On behalf of both CDC and also the Department of Health and Human Services Working Group on Lyme and other tick-borne diseases, I want to welcome you today to our special webinar on vaccines for Lyme disease, past, present, and future. I'd like to begin just by emphasizing that the views expressed in this webinar are those of the participants and do not reflect the official policy or position of CDC, the Department of Health and Human Services, or the U.S. government. As many of you will know, Lyme disease is the most common vector-borne illness in the United States. In 2013, there were over 36,000 cases reported, making it the fifth most common reportable disease in the U.S. These are reported cases. The actual number of cases occurring each year is estimated to be around 10 times this number. Since becoming nationally notifiable, Lyme disease has been increasing steadily in the U.S., both in case numbers and in geographic distribution. Consequently, identifying and validating effective prevention and control tools and methods is one of the highest priorities at CDC. The primary objective of our webinar today is to review the history of human vaccines for Lyme disease and discuss the prospects for next-generation human Lyme disease vaccine. We have 4 formal presentations, followed by a summary and conclusion, after which we'll take time for questions and answers. The topics and the speakers are the following. The first is why we need a vaccine for Lyme disease, Dr. Stanley Plotkin. The second is potential vaccine strategies and targets for Lyme disease, Dr. Rich Marconi, followed by a critical appraisal of LymeRix and lessons learned, Dr. Robert Aronowitz, and then progress toward a second-generation Lyme vaccine, Dr. Nina Resnick, and then finally the summary and conclusions by Dr. Larry Madoff. Questions and answers will be answered following the last presentation and can be submitted online through the webinar interface. Please provide your identity and indicate the speaker to whom you're addressing your questions. For those of you who are unable to join us today for technical reasons, you can see all these presentations in their entirety. They'll be posted at our website, both the audio and video portions, at the website you see on this slide, www.cdc.gov/Lyme. So with that, I will introduce our first speaker. Our first speaker is, the title is Why We Need a Vaccine for Lyme Disease, given by Dr. Stanley Plotkin. Dr. Plotkin is Emeritus Professor of the University of Pennsylvania. Until 1991, he was a Professor of Pediatrics and Microbiology at the University of Pennsylvania, Professor of Virology at the Wistar Institute, and at the same time, Director of Infectious Diseases and Senior Physician at Children's Hospital of Philadelphia. For 7 years, he was Medical and Scientific Director of Sanofi Pasteur and is now consultant to vaccine manufacturers and nonprofit research organizations. He is a member of the Institute of Medicine of the National Academy of Sciences and the French Academy of Medicine. His bibliography includes over 700 articles, and he's edited several books, including a textbook on vaccines. He developed the rubella vaccine that's now in standard use throughout the world. He's co-developer of the newly licensed pentavalent rotavirus vaccine, and he's worked extensively on the development and application of other vaccines, including anthrax, oral polio, rabies, varicella, and cytomegalovirus. Dr. Plotkin?
Yes, good morning. So let me tell you, first of all, that I have no financial interest in Lyme disease vaccines, but after the withdrawal of a vaccine, which we'll talk about later, I became rather incensed about the fact that we had no way of preventing Lyme disease vaccine. Now, the next slide is a case history of a person named Alec P. He was living in Chester County, Pennsylvania, near Philadelphia. He walks his dog a lot. At one point, he developed an erythematous rash behind his knee. He went to a physician, but his physician thought it was cellulitis and treated it locally. The result was that one day he was out with his dog and collapsed and was unresponsive. The neighbor called 911. A medical technician came and measured a heart rate of 35 per minute in Alec. He was taken, of course, to the local hospital where an experienced cardiologist who had seen many cases like this before suspected Lyme disease. So Alec underwent insertion of a cardiac pacemaker and was started on intravenous antibiotic. It so happened that Alec P. is my son. So that, of course, made me even more incensed about the situation. Now, the advice to people currently to avoid Lyme disease, as illustrated by an article in the Philadelphia newspaper, is to avoid wooded areas, walk on trails only, use DEET repellents, treat clothing with permethrin, shower within 2 hours after walking, do body tick checks, check pets for ticks, and dry clothes in high heat. Well, that's fine. But, of course, how many people are going to do all of these things? And even then, there are cases of Lyme disease. So what's to be done? Well, there was a vaccine that was developed in the 1990s. And as I'll show you in a moment, there was a permissive recommendation by the CDC for vaccination of adults with the vaccine. Adults because the manufacturer had not shown data in children. It was withdrawn by the manufacturer in February 2002. It was withdrawn because the sales had declined, but also because there were class action lawsuits against the vaccine, arguing that the vaccine was causing harm. In retrospect, the FDA's analysis of the data were negative. The scientific analysis of the data concerning the putative reactions showed that there was no scientific reason for believing that the vaccine was causing harm, but nevertheless the vaccine was withdrawn. And this was the first time in the modern era that an FDA-licensed vaccine was withdrawn. So this precluded achievement of any goals to manage Lyme disease, to prevent it. Now, my argument is that we should have a vaccine because of the numerous cases in the U.S. And by the way, it is also a disease in Europe where there is an estimated 85,000 cases per year. Also, vaccination with the original vaccine was shown to work at a level of 80% or better. So it was not an ineffective vaccine. And other preventive measures have not been shown to work on a public health scale. This slide simply shows data from the trial that licensed the vaccine, and you can see that, as I said, the efficacy overall was 80%. Now, it's important to note that the recommendations made by the CDC were rather tepid, and the comments made after licensure also at the time were rather tepid, illustrated here, that this is a vaccine for yuppies, etc., ignoring the wide prevalence of the disease in the Northeast and also the upper Northwest. So the recommendations for Lyme disease vaccine illustrated here were, and the first recommendation is the most important, that it should be considered for persons aged 50 to 70 years who engage in activities that result in frequent or prolonged exposure to tick-infested habitat. So certainly not a very strong recommendation, which is one of the reasons the vaccine was not used. So many mistakes were made with the original vaccine. The manufacturer advertised directly to the public without convincing physicians of the importance of vaccination, and it is recommendation by physicians that determines whether the public uses a vaccine or not. They launched a pediatric study too late, and by the way, the pediatric study showed that the vaccine would have been even more effective in children than in adults. And the manufacturer also failed to engage patient groups interested in Lyme disease, patient groups who are concerned about chronic results of infection by the bacterium. CDC, in my opinion, made a mistake by assigning the vaccine recommendations to staff that had no direct experience of Lyme disease. That is, staff that were not stationed in the Northeast, and making weak recommendations. There was weak support by opinion leaders, there was opposition by patient groups, and as I mentioned, the class action lawsuit. Well, so what's the situation now? There is the opportunity for a vaccine, or really multiple vaccines, against Lyme disease. The Baxter company in Austria, and we'll hear more about that, have developed a new vaccine which is very similar to the old one, but it takes away, it deletes from the proteins in the vaccine that which was suspected to be the cause of reactions. And again, I emphasize that that suspicion was not confirmed. Nevertheless, out of caution, this putative cross-reactive protein, or part of the protein, has been removed. So that is a possible vaccine, although unfortunately that manufacturer has gone out of business. This slide simply shows that the new vaccine would have been effective against not only the strain of bacteria in the U.S., but also strains present in Europe. There is another company in Austria called Valneva that is developing still another vaccine based on the protein of the bacteria called OspA. And we'll hear more about the proteins in the bacterium that could be protective when used as vaccines later on in these presentations. At any rate, that is a second possible vaccine that could be developed based on OspA. But I want to emphasize, and again we'll hear more about this, is that there are many other possibilities for developing vaccines against Lyme disease. This slide, which is really not complete, but it just shows that there are many other potential proteins that could be used in a Lyme disease vaccine. And so there are many ways in which scientists could develop ways of protecting the public against Lyme disease. In terms of cost effectiveness, which is an important issue in modern times, considering the economy, et cetera, there were several cost effectiveness estimates made when the first vaccine was available. And they're listed here, and I'm not going to go into any details, but what they show is that by current standards, a Lyme disease vaccine would be cost effective because of the numerous cases, because of the medical care that's required, because of the possibility that there is a chronic Lyme disease, that there are symptoms of infection after treatment for Lyme disease. So for all of those reasons, economic as well as public health and medical, there should be a vaccine against Lyme disease. So here is what I would call the profile, the target product profile for a new Lyme vaccine. It should protect against all three major Borrelia species, both those in the U.S. and Europe. It should remove the putative cause of reactions, which the modern vaccines do. It should be given to children as well as adults with the prospect that the vaccine will be very highly effective in them. It should overall in adults have an efficacy of more than 80% after 3 doses, and perhaps fewer doses could be used. It should have prolonged effectiveness at least for 2 years and perhaps longer. And of course, the vaccine itself should not cause arthritis as the disease does. So my conclusions are that there is a great need for vaccination against Lyme disease, both here in the U.S. and in Europe. The OspA vaccine, the OspA vaccine, the original vaccine was a scientific success, but a public relations fiasco. But fortunately, new OspA vaccines and also vaccines made with other proteins are feasible. And many of these antigens are likely to work to prevent Lyme disease. So this is a public health mistake that should be corrected as soon as possible. Thank you.
Thank you, Dr. Plotkin. And with that, we'll move on to our next speaker. And while loading the slides here, the next talk is potential vaccine strategies and targets for Lyme disease, and will be given by Dr. Rich Marconi. Dr. Marconi is a professor in the School of Medicine at Virginia Commonwealth University Medical Center. He earned his PhD at the University of Montana and then conducted his postdoctoral training at the Roche Institute of Molecular Biology and at the National Institutes of Health in Hamilton, Montana. Dr. Marconi has 28 years of experience in the study of pathogenic spirochetes. His current research interests are diverse and include the study of molecular mechanisms of pathogenesis for Lyme disease, tick-borne relapsing fever, and periodontal disease. His laboratory has demonstrated experience in product development and has patented an innovative chimeric vaccine for Lyme disease. He is an editor for the journal Pathogens and Disease and has served in numerous other professional capacities, including president of the Virginia branch of the American Society of Microbiology, a chartered member on the Vaccine for Microbial Diseases NIH Study Section. To date, he's published more than 90 high-impact peer-reviewed research articles, books, and chapters and reviews. So, Dr. Marconi.
Thank you, Ben. I appreciate the opportunity to speak with everyone today. So today I'm going to tell you about our work, which is focused on new vaccine strategies for Lyme disease, and I'm going to introduce a new term and a new technology that we're developing that we refer to as Chimeritope vaccines. But first, by way of background, I want to talk a little bit about how vaccines for Lyme disease might work. And there are several different categories of vaccines that can be pursued, like the OspA vaccine that Dr. Plotkin talked about. That was primarily a transmission-blocking vaccine. So that's a vaccine that's served to block the passage of spirochetes moving from ticks into mammals. That type of a vaccine its protection is dependent upon pre-existing antibody and requires frequent boosts. Another category or approach that can be taken are vaccines that will clear an infection. And this is a fairly traditional vaccine approach where protection is driven by immune memory response as opposed to pre-existing antibody. This approach requires fewer boosts. In addition, there's a lot of work going on with bait vaccines in which people are pursuing paths to decrease the carriage of Lyme disease spirochetes in nature by essentially vaccinating through oral immunization mammalian reservoirs of the disease. And lastly, there are tick vaccines, which exploit different components of the tick, and these vaccines are designed to inhibit tick feeding and pathogen transmission. Again, this is an approach dependent upon pre-existing antibody. Some important considerations that need to be thought about when pursuing a vaccine candidate. We need to know at what stage in the enzootic cycle or stage of disease that particular protein is produced by the bacteria. This is important because it will influence the protective mechanisms that come into play, and will also influence vaccination schedule. Lastly, we need to consider whether the vaccine protein, whether its sequence varies among strains, and it is, of course, essential that we design a vaccine that will protect against all Lyme disease spirochete species and against all strains. I'd like to use an animation to highlight the idea of differential expression of Lyme disease spirochete proteins during the course of the enzootic cycle. Here we see a cartoon depicting a tick that is ready to feed with spirochetes in its midgut, and you can see by the color coding that it's producing high levels of outer surface protein A. Once the tick begins the blood meal, the blood enters into the tick, and the spirochetes transition from producing OspA to producing Ospc. And then the spirochetes will pass through the salivary glands and into the mammal, where they'll maintain Ospc expression for weeks to months before Ospc expression is downregulated. So when the tick detaches, Ospc will with time become downregulated, and there will be a transition to other outer surface proteins, one of which is outer surface protein E. So the technology that we're working on in our lab is what we refer to as Chimeritope technology, and I think it's a new direction for Lyme disease vaccines. To define it for you, a Chimeritope is a protein comprised of a series of short, well-defined, protective linear epitopes that can be derived from one or more proteins or protein variants. One of the positives about this approach is that regions of a protein that are non-productive or that elicit adverse events can be eliminated. In addition, by utilizing solely linear epitopes, we can eliminate the need to maintain conformational epitopes. And as we are trying to do, a Chimeritope can be designed to work through multiple synergistic mechanisms of protection. For example, by including epitopes derived from both OspA and Ospc, one vaccine can serve to both inhibit transmission as well as clear bacteria that do in fact enter into the host. One of the challenges that I alluded to with outer surface proteins of the Lyme disease spirochetes and vaccine development is their inherent genetic diversity. And while I won't go into discussion of this in detail, I'll tell you that analyses from several labs have shown that there are approximately 30 different phyletic types or variants of Ospc that exist in nature. An individual strain of the Lyme spirochetes, however, will express only a single Ospc type protein. However, ticks are infected with a mixed population. So when a tick bites, it can transmit strains that are producing a series of different Ospc types. So this is something that needs to be addressed in an Ospc-based vaccine. We have to account for Ospc variation. OspA is somewhat less variable, but also has significant variation, and there are 6 major serotypes of OspA that have been defined. So the problem is, how do we overcome variation in OSP proteins and design a vaccine that will convey broad protection? And the path that we are pursuing, once again, is the development of Ospc and OspA Chimeritopes. So to put in context for you the variation in Ospc and how it influences immune responses, I'm going to show you some data from a paper we published several years ago. And in this experiment, along the top panel that you'll see on the figure on the left, you'll see a series of proteins that are appearing, and these are recombinant Ospc proteins of several different Ospc types. And then in the images that are underneath it, you will see that there are a series of immunoblots in which we screen these recombinant proteins with serum from mice that were infected with strains that express Ospc of a known Ospc type. So just focusing on the type A panels, which are indicated there, you note that serum collected from a mouse that was infected with a type A Ospc after 2 weeks of infection reacts exclusively with type A Ospc. After week 8 of infection, that serum remains highly specific for type A Ospc and does not react with other variants. So again, it highlights the type-specific nature elicited during infection towards Ospc. I'll skip the remaining data on the bottom and draw your attention to the right-hand panel, which shows a molecular structure, a model for Ospc. Ospc is a dimer, and we can see the dimer in this particular image, and it's color-coded. The red residues or red parts of the protein are highly conserved, whereas the blue to purple parts of the protein are variable. The fact that Ospc elicits type-specific responses indicates that it's these blue or purple or variable regions of the protein that are driving the early immune responses to this particular protein. So now with an understanding of this variation in hand, we sought to pursue an approach where we could potentially link a series of epitopes derived from diverse Ospc proteins into a single molecule to make a more broadly protective protein. And to do that, we had to identify the location of the epitopes in the Ospc molecule. In the left-hand panel, you'll see an image which depicts a series of lines, basically, and those are denoting different subfragments of Ospc that we made as recombinant proteins. And I will keep a long story short with this data and advance to the end of this animation to make the key point, and that is that if you look at this animation, you'll see two regions that are highlighted in red. Those represent two linear epitopes that we identified in the Ospc molecule that we designated as the loop 5 and the alpha helix 5 epitopes. And the location of those molecules on an Ospc monomer is shown to the right, and you can see that they exist on the surface of the protein. Okay, so with that, we sort of entered into the world of developing what we, again, call the Chimeritope approach. And the first thing that we sought to do was to first establish that these epitopes that we identified, when delivered into animals, will in fact elicit antibody that will kill the bacteria, which is called bactericidal activity. So mice were vaccinated with the type A loop 5 epitope. Serum was harvested, and that serum was then incubated with an Ospc, with a strain producing an Ospc type A protein. And what we're looking for in this assay is to determine if the antibody can destroy the cell and lyse them. And if you look at the right-hand panel, cells that survive exposure to antibody stain green, and those that are killed will stain red. And you can see that when the antiserum is delivered with complement, you get significant killing of the spirochetes. So the next step then was the genesis or the beginning of the development of a Chimeritope. And we took the loop 5 and alpha helix 5 epitopes from 4 different Ospc proteins, types A, B, K, and D, and assembled a chimeric protein that now has 8 different epitopes in it. And that protein was tested in mice to determine if it would elicit bactericidal antibody and kill bacteria. And that is, in fact, what we saw. We found that vaccination with that protein induced very efficient killing of strains of different Ospc types. Okay, so to highlight how we sort of evolved in developing these Chimeritopes, this particular slide shows you 8 different molecules of Ospc, and they've been color-coded to indicate that they're different variants, and the region that we've identified as important in protective responses is highlighted with the different colors. What we have done is we took the regions of interest from these proteins and simply assembled them into a new protein. So this is not an Ospc protein, but instead it is a new protein that consists solely of epitopes derived from Ospc. And with that background in mind, I'm going to focus and talk about some data that we've generated with a new series of Chimeritopes. These are called the CHV through CHV3 series. And these Chimeritopes, the 3 that I'll talk about, all possess the same loop 5 and alpha helix 5 epitopes derived from 9 different Ospc types. So they have essentially 18 individual epitopes incorporated into the constructs. These constructs were generated and were tested in a canine model for protective efficacy. I'll tell you a little bit about each construct. CHV1 has loop 5, helix 5 epitope pairs included in it that are separated by linker elements, just short sequences or short segments that will separate each individual epitope. CHV2 has the same epitope order as CHV1, but we eliminated the linkers, and that's advantageous because it means we can pack more into a molecule of the same size. CHV3 has a different order of epitopes, and there are no linkers present. So we tested these 3 Chimeritopes. We also tested one of these Chimeritopes in combination with OspA, full-length OspA, and a vaccine cocktail. And each of these was administered to dogs. There were 8 dogs per group. The vaccine was delivered at 0 and 21 days, and then a tick challenge was conducted at day 42. And of course, we determined prior to doing this the optimal vaccine dose, and we verified that when these constructs were put into canines, that they would all elicit productive antibody responses that would detect all the component epitopes in the vaccine. The next step was to determine if vaccination with these proteins elicits antibody that can kill the bacteria. And I'm going to present some select data here on just 3 of the groups, and that's the CHV2 group, the combination group, and then OspA alone. And these are in vitro assays in which we take the serum from the vaccinated dogs and we determine if it is efficient at killing the Lyme disease spirochetes in the laboratory. And as you can see, what we are looking at is percent killing as assayed by dark field microscopy. And each of these groups, and a serum collected from each dog, was very effective at destroying spirochetes in the laboratory. The other constructs I alluded to also were quite effective in eliciting potent bactericidal antibody. All right, so the next step then was to determine how this vaccine is actually working in canines. And specifically, we looked to determine if vaccination would block Lyme disease-associated histopathology. So the different groups that were analyzed are shown on the right. And basically, we collected 88 samples from each dog, 88 histopathological samples, and they were analyzed blindly, and then we evaluated the frequency with which lesions developed in canines vaccinated with each group. Now, the control group, shown on the top, is the T01 group. That's a non-vaccinated group of dogs that were infected via tick bite. And if you look at that data, you'll see that for that particular group, 55% of the lesion sites, or the sites that were analyzed, displayed lesions consistent with Lyme disease. If you look at the table on the left, you'll see that CHV1 through CHV3 all dramatically reduced the percentage of sites that were determined to have Lyme disease lesions, with CHV2 being the most effective. OspA was also effective at reducing the number of lesions that occurred, but the combination of CHV2 with OspA was rather dramatic and completely blocked the development of any lesions in these dogs. The CHV2 Ospc Chimeritope was particularly effective at decreasing the level of joint involvement, whereas with OspA, while OspA elicited bactericidal antibody, many of the dogs still displayed joint pathology. However, as I said a moment ago, both together were very effective in reducing histopathology in the dogs. Okay, so in this slide, we then sought out to make a Chimeritope that's based on OspA, with the idea being that an Ospc Chimeritope and an OspA Chimeritope would make the ideal combination that would completely block Lyme disease development in mammals. So using the approach that I outlined earlier, we conducted epitope mapping analyses, and this figure on the left shows you the different fragments of OspA that were generated, and they were then screened with serum from infected mice, which is shown on the right. There's one particular fragment that spans a region from 221 to 240. Those are the amino acids in the protein. That was detected by virtually all of the animals that were infected, with the exception of one, which is one mouse, M6. So we were able to identify a linear epitope in OspA, and this was very exciting to us because it raised the possibility that we can now incorporate OspA into our Ospc Chimeritope, or make 2 Chimeritopes, one for Ospc and one for OspA. In the next slide, we show you some very important data. And in this experiment, we took that particular epitope, the 221 to 240 epitope, and we delivered it into mice, and then we assessed whether the antibody that was elicited has bactericidal activity. An important control is shown all the way to the right, and that's antisera generated against full-length OspA, which is very effective at killing. If you look all the way to the left, you'll see that the peptide itself was also very effective at eliciting killing, nearly 85%. So this indicated to us that we do not need the full-length OspA protein to elicit productive antibody responses. This epitope also elicited good killing activity against strains from Europe, including isolates of Borrelia bavariensis and Borrelia salii. That killing was not complete, however, so clearly we need to incorporate additional variants of this epitope into a new Chimeritope. And on the right, you'll see a schematic or structure of OspA. The region indicated in red is the 221 to 240 epitope. You'll see where it's located in the molecule. And the boxed in area on the top shows you the region of a previously identified conformational epitope in OspA called the LA2 epitope. Again, our goal is to get around the use of conformational epitopes, and I believe this data suggests that that is possible. So to summarize and talk about future directions, we have shown and identified linear epitopes within Ospc that are called the loop 5 and Helix 5 epitopes. We've demonstrated that they elicit early antibody responses. We have shown that Ospc Chimeritopes are immunogenic and well tolerated in animals. We have done extensive analyses of epitope ordering in these constructs and in optimizing their design. And we have demonstrated that Chimeritopes can in fact elicit bactericidal antibody and significantly reduce histopathology in animal models. CHV2 of the set that I just showed you proved to be the most effective and was particularly good at reducing joint pathology. Canines that were vaccinated with CHV2 and recombinant OspA did not develop any detectable pathology, and in addition, there was no seroconversion that occurred in these canines after challenge. Similarly, we did not detect any PCR positive or culture positive data after challenge. I've also shown you that an OspA linear epitope has been identified and that that epitope induces very potent bactericidal activity. So our goal in moving forward is to develop Ospc and OspA Chimeritopes that are specifically tailored for humans. By that I mean that are designed to prevent infection with the strains that commonly infect humans. In addition, we are seeking to develop a multi-pathogen Chimeritope vaccine. This would be a vaccine that can protect not only against the multiple species that cause Lyme disease, but also other tick-borne pathogens like Anaplasma and Babesia. So we believe that this is a very powerful new technique, and we're also excited about the potential of using Chimeritopes as powerful new tools in Lyme diagnostics. And with that, I'll show you, I want to acknowledge the people who were involved in the study, and a full disclosure is presented on the right. And I thank you very much for your attention.
Thank you, Dr. Marconi. So our next talk is entitled A Critical Appraisal of Limerick's and Lessons Learned, and it will be given by Dr. Robert Aronowitz. Dr. Aronowitz is Professor and Chair of the History and Sociology of Science at the University of Pennsylvania. He studied linguistics at Berkeley before receiving his MD from Yale. At Penn, Aronowitz was the founding director of the Health and Societies Program and co-director of the Robert Wood Johnson Foundation Health and Society Scholars Program. He is the author of Making Sense of Illness, Science, Society, and Disease, Unnatural History, Breast Cancer in American Society, Risky Medicine, Our Quest to Cure Fear and Uncertainty, and he's co-editor of 3 Shots at Prevention, the HPV Vaccine, and the Politics of Medicine's Simple Solutions. He's also published widely on the history of medicine and disease. So, Dr. Aronowitz?
Thank you, Ben. So I approach this topic as a historian and physician, not as someone engaged in either the basic science or clinical research. And my historical observation really boiled down to 2 points on this slide. First is that the failure of the first-generation Lyme disease vaccines that Dr. Plotkin alluded to is one chapter of the nearly 40-year-old controversy over the definition and diagnosis of Lyme disease. And the second point is that if the parameters of that controversy, of that underlying controversy, are not understood or even completely understood, new vaccines may become entangled in the same problems once again. Let me review very cursorily, that is, the history of the Lyme disease controversy, trying to emphasize the answers to 2 questions. The first being, why did the definition and diagnosis of Lyme disease become so controversial? And secondly, what is at stake in these controversies? And I don't use a particular historical method, but an interpretive stance, which I'm calling framing disease, whose important elements are the following. There are no hard and fast rules for recognizing a new disease. What often counts as a new disease often depends on values, interests, and distinction historical events. Biological processes place limits on how diseases can be defined, but how a disease is defined often results in winners and losers. The main historical question is why Lyme disease was considered a new disease, and to explore the consequences of being framed that way. And to simplify a very complicated history, attention and later controversy followed from a 1976 study that claimed the appearance of a new disease. First it was called Lyme arthritis, then Lyme disease. Erythema migrans was called the rash of Lyme disease, minimally acknowledging the much older history of erythema migrans in Europe, which over the previous half century evolved to be understood as a protean disease that was likely caused by bacteria carried by ticks, and after the introduction of penicillin after World War II, responsive to antibiotics. And there was so much understood as a new disease, the European cases of erythema migrans were renamed Lyme disease with some resistance in Europe. Okay, so being labeled a new disease had consequences. In the early years of the Lyme disease investigation, viruses, not bacteria or spirochetes, were prime etiological suspects. And many of the initial cases were, despite what was known about erythema migrans, were not treated with antibiotics. But most importantly, the newness of the disease brought fear, attention, and contributed to the controversy over the proper definition, diagnosis, and treatment of Lyme disease. In other words, what's now understood as a chronic Lyme disease controversy. What is this controversy? Well, to simplify greatly, overall, all expert diagnostic criteria result in doctors, not patients controlling the diagnosis. And I'm going to caricature here two perspectives, the orthodox and the heterodox. The orthodox being that of the most experts in Lyme disease, who believe that Lyme disease is easy to diagnose on clinical grounds early. Late manifestations are relatively rare and must be one of the few narrowly defined conditions. Short courses of antibiotics are generally effective for most cases. And I don't want to go into it at the moment, but the heterodox perspective is almost a mirror image of this orthodox position. Okay, the controversy, let's fast forward to the 1990s with Lyme disease vaccines. Where the controversy continues. And Dr. Plotkin mentioned a few of these things, so I'll go very quickly here. Two OspA vaccines were developed. Vaccines that were developed, unlike what the new ones Dr. Marconi was describing, block transmission within the tick vector. The efficacy and safety of these vaccines was generally well established in two clinical trials. One vaccine was never submitted to the FDA. Lymerix, which did get FDA approval and endorsed, is tepidly by the ACIP, but after a large direct-to-consumer advertising campaign and 2 years of less than expected sales and lawsuits, the vaccine was withdrawn. The pre-existing Lyme disease, my main point here is the pre-existing Lyme disease controversy shaped the vaccine controversy. Some in this heterodox community initially embraced, but then later became opponents of the vaccine. One expert Lyme disease clinician observed early during the controversy that, and I quote, most of the lobbying is focused on what Lyme disease is rather than how to prevent it. The diagnostic criteria in the trials were contested for being too narrow, too much like the orthodox view. There was upset over changed criteria for Lyme disease laboratory diagnosis in advance of the vaccine introduction. There was in 1994 some Dearborn criteria that removed the Western blot that might have caused false positive diagnoses of Lyme disease after vaccination. The main argument of the skeptics was that the vaccine caused a Lyme disease-like disease whose mechanism was molecular mimicry to the antigen used in the vaccine. why was the vaccine controversial? And here I'm highlighting things beside any belief in the danger of the vaccine. It was, as Dr. Plotkin said, it's not generally accepted by most scientific experts. First, evidence of the vaccine's efficacy and safety appeared to reinforce the orthodox definition of the Lyme disease used in clinical trials. In other words, once the vaccine's efficacy was established, there was a collateral implication that the narrow diagnostic criteria used to establish this efficacy worked as well. Competing claims of vaccine-induced Lyme disease symptoms reinforced the heterodox view of immune mechanisms of chronic Lyme disease. These two universes are well illustrated by the incommensurate interpretations of lack of asymptomatic seroconversion. And what I mean by that is that rather than understand, and this is the evidence from the trial conducted by Alan Steer, rather than understanding the absence of asymptomatic seroconversion among the vaccinated as evidence of vaccine efficacy, some detractors offered a diametrically opposed interpretation. The vaccine caused people with prior or latent infection to become symptomatic with Lyme disease, or the Lyme disease-like disease, resulting in no one left to be symptomatic. The regulatory bodies concerned with threats to trust in mandated vaccines by niche vaccines led to those type of recommendations that Dr. Plotkin mentioned. And basically, what was at stake here was not just the yuppie disease, but a target disease that was treatable, where there was no herd immunity going to be induced by vaccination. There were other ways to prevent. And the core issue remained the legitimacy of chronic Lyme disease. So one puzzling question here is, why did the skeptics of the vaccine beat the Lyme disease medical establishment and the pharmaceutical industry? And this seems puzzling at first, given the resources for marketing, the widespread concern about Lyme disease, the evidence of efficacy in the trials, and many of the points Dr. Plotkin mentioned. And the first point, obviously, is the public health case for vaccine was limited relative to the things, to the case for our mandated vaccines. But a more subtle point, I think, is that the market success would depend on widespread concern and fear, conditions consistent with the heterodox perspective. And I've been concerned that a product whose market success depends on selling fear and detention to disease can itself be easily undermined if associated with danger itself, which is what happened in the course of the Lyme vaccine controversies. And of course, other issues that have been mentioned by many observers, the cost of the vaccine, the need for boosters, the weak recommendations. This is just a picture of something I found during the initial vaccine introduction in Philadelphia, where I live, that was trying to spread awareness, but also depended on spreading some degree of fear of Lyme disease. So what are the lessons to be learned? And I'll just make two sets of observations. One about the Lyme disease controversy itself. I think we need to acknowledge the legitimacy of suffering, whether a known and agreed upon mechanism exists or not. And I think it's also possible to try better at accepting a diversity of diagnostic names and possible natural histories and decouple the name and diagnosis issue from treatment decisions. And we could all do with less hubris. What we call Lyme disease is, in the end, a choice, constrained but not totally determined by biological and clinical realities. For the next generation of Lyme disease vaccines, I think we need a greater awareness at each point of testing and marketing of this pre-existing Lyme disease controversy. And in particular, I think we need caution over the marketing and other initiatives that depend on selling fear of disease. And at the same moment, I think we need honesty and humility about the limited niche that any vaccine is likely to occupy. Thank you.
Thank you for that presentation. And now we'll go to our next speaker. Our next talk is Progress Towards a Second-Generation Lyme Vaccine. It will be given by Dr. Nina Resnick. Dr. Resnick has a background of more than 10 years in biotechnology, vaccine development, and immunology, both in Austria and in the U.S. In her latest function as Manager of Clinical Development of Vaccines at Baxter Bioscience in Austria, she was responsible for the strategic assessment and planning of top strategic projects, development of clinical research initiatives, and related global scientific communications. She developed global clinical strategies for Lyme borreliosis vaccine, advancing it from the preclinical stage to clinical phase 3 readiness. She also had key roles in vaccine efforts with the Ross River virus vaccine and with seasonal influenza vaccine development. Prior to joining Baxter, she developed live attenuated influenza vaccine candidates based on the reverse genetics technology and moved them from bench to clinical stage at ABIR Green Hills Biotechnology in Austria. Prior to a career in pharma and biotech industry, she worked at Mount Sinai School of Medicine in academic virology, developing new approaches for live attenuated influenza vaccines. Dr. Resnick holds patents in the field of vaccine development and is the author and co-author of numerous peer-reviewed publications and recipients of numerous rewards. So, Dr. Resnick?
Thank you very much. I'm honored to present you the recent developments towards multivalent recombinant of the Lyme borreliosis vaccine or Lyme disease vaccine, which has been developed by Baxter, as was already mentioned. So before I present you with the data, I want to give you just a little bit of a background information. I'll keep it short, as you have already heard parts of it in the previous talk. So the candidate vaccine is based on the outer surface protein A, which has been proven in 2 previous phase 3 efficacy trials of monovalent OspA vaccines in the U.S. to be a protective antigen. But as you also already heard as well, despite these 2 successful phase 3 trials, there were some issues associated with these vaccines. First of all, the OspA type 1 contains an epitope mimicking the human lymphocyte function-associated antigen 1, which was reportedly associated with human arthritis following infection of persons with a specific HLA type. Although, as you also heard by Dr. Plotkin, no evidence supported the hypothesis, and a subsequent study by Dr. Steer suggested that molecular mimicry does not have a role in Lyme arthritis, this association was one of several factors that contributed to poor acceptance of the vaccine by the public as well as by the physicians. And secondly, OspA, as you already heard as well, is antigenically heterogeneous, which means that a multivalent vaccine is required to generate broad-based coverage in Europe. So the strategy to address these issues was now to develop a trivalent chimeric antigen formulation, which would provide full coverage of all strains responsible for human disease in the U.S. as well as in Europe. And as I will show you on the next slide, this approach also allows for the deletion of the putative aerugenic HLFA1 epitope. I'm aware that you have seen this figure as well by Dr. Plotkin, but I just want to mention here that with the knowledge of the structure and function of OspA, it was possible to generate the chimeric OspA molecule. And I'll show you here an example of type 1 and type 2 strains. On the left, you see 2 native OspA molecules of serotype 1, the Burgdorf resensus stricture, which is the only borrelia species causing Lyme borreliosis in the U.S. And next to it, in red, you see the serotype 2 corresponding to the Burgdorf abscali, which is the Borrelia strain predominant in Europe and also has a tropism for skin manifestations. By the way, and that is all taken from Lydia Dahl from CID in 2011. So you can also see here the major protective loops, A1 to A3, as well as the OspA epitope mimicking the HLFA1 sequence in the type 1 strain. The strategy that was now used fused the proximal and the distal parts of the 2 different OspA antigens to generate a recombinant OspA antigen, which you can see on the right side. And importantly, the overall 3-dimensional structure of the native OspA molecules and the key protective epitopes were maintained. And as already mentioned, this approach allowed to replace the putative cross-reactive OspA1 epitope with the corresponding OspA2 sequence, and you can see that on the right side indicated by the red arrow. So this here, you can see, are just the one chimera, And for a whole vaccine formulation, you need additional chimeras. So what was done in addition was that the same approach was used for OspA serotype 6, the Bacterini strain, and for the Bavariensis strain, as well as for 5 and 3, the 2 Garini strains, resulting then in a vaccine formulation containing 3 novel antigens of OspA. And with this together, that would not only provide coverage against the major disease-causing Borrelia species, but also against the major disease syndromes in the U.S. and also in Europe and potentially throughout the world. What I want to, again, briefly mention to you, although we have heard it by Dr. Marconi, is the mechanism of A-mediated protection. So following attachment of the tick to the host, there are 2 important things to remember, which are, first of all, OspA is only expressed when a spirochete is present in the tick midgut, and prior to transmission from the tick to the host, OspA expression is turned off and OspC is instead expressed. Now, in the case of a tick feeding on an OspA immunized host, the tick during the blood meal takes up OspA antibodies from this immunized host, and these antibodies bind then to the spirochetes in the tick midgut and either directly kill now the bacteria through bactericidal antibodies, or the antibody blocks the attachment to receptors on the tick gut epithelium, which then results in failure of the spirochetes to migrate into salivary glands, and thereby the transmission to the OspA immunized host is blocked. So again, that's the mechanism of a transmission blocking vaccine. And that brings me now to the implications for an OspA vaccine. You have to keep in mind, it's very crucial for that, and I think it was also mentioned before, is that for protection it is essential that high levels of circulating antibodies are present in the blood. So let me jump now into the data that was generated in a phase 1/2 clinical study. And I will mainly spend my time talking about the first part of the study, investigating the multivalent vaccine in a dose escalation double-blind manner in a healthy adult population, and that has been a seronegative population. And I will also briefly mention to you the results of the second part of the study, and we're in an adult population that with this population has been previously infected with Borrelia burgdorferi sensilato, and I will mention that only in the summary and conclusion slide. So just for you to understand how the study was designed, I'll briefly introduce you to the study protocol. So it was a safety and immunogenicity study incorporating different parts, or we call them sections. So I'll focus mainly, as I already said, on section 1, which was the dose escalation part, the unprimed seronegative subjects were aged 18 to 70 years. 3 dose levels were investigated, so 30, 60, and 90 micrograms with or without aluminum hydroxide as a measurement. Overall, 300 participants were included, so 50 participants per study arm. The immunization schedule consisted of 3 vaccinations at monthly intervals at day 1, day 29, and day 57. And selected study arms received a booster at 9 to 12 months. And I tried to schematically illustrate that at the bottom of the slide, where you see the priming, the surface resistance, and the booster part. The little fire ranges indicate the vaccinations, and the blue, white, and red stripes underneath indicate the blood draws that have been taken. And as said, the endpoints were safety, and here specifically the adverse events within 7 and 28 days after each vaccination, as well as immunogenicity for each of the 6 individual serotypes as measured by ELISA, as well as functional assays such as surface binding assay or killing assay. So after all the introduction, I'll start now with the safety data. Here I show you the percentage of participants in the different dose groups in the adjuvanted on the left side and non-adjuvanted formulation groups on the right side with solicited local reactions, that's the bottom at the top part, and systemic reactions at the bottom within 7 days of the first vaccination. And with respect to local reactions, the most common ones were injection site pain and tenderness, and you can see that those are the last 2 bullet points in the local reaction section. And what I also want to mention here that there was no statistically significant difference between the adjuvanted and the non-adjuvanted formulations when you look at the local reactions. But when you look now at the systemic reactions, there are clearly malaise, fatigue, and headache were the most frequent systemic symptoms, and were also more frequent in recipients of the non-adjuvanted compared to the adjuvanted formulations. And importantly, when we were conducting a statistical analysis of this data, the frequency of systemic reactions and the risk for moderate or severe systemic reactions was significantly lower for the adjuvanted compared to the non-adjuvanted formulations. And also another thing was that there was no difference between the 30 and 60 microgram adjuvanted formulations with respect to systemic reactions. Finally, the majority of systemic reactions occurred within 24 hours after vaccination, and no vaccine-related serious adverse events were reported. Coming to the next slide, you see here the percentage of participants with systemic reactions. I intentionally now am not presenting to you the local reactions because, as I mentioned before, there's not a big difference between the adjuvanted and non-adjuvanted formulations. So I'm showing you systemic reactions within 7 days of each vaccination, so at the first, second, and third, as well as after the booster vaccination, that have been rated by the investigator to be mild, moderate, or severe adverse events. And what you can see here, I mean, actually you cannot see here, it's something I'm going to tell you, is that most adverse events occurred within 24 hours of immunization. They were mild in severity and resolved spontaneously within 72 hours. And what you also see is that successive vaccinations were generally associated with a decrease in the risk of systemic reactions, and that is important now that the adjuvanted formulations are better tolerated compared to the non-adjuvanted formulations. Another thing I want to mention is that the participants in the 90 micron, just in case you're wondering, so the participants in the 90 micron non-adjuvanted dose groups had the most systemic reactions. Many of them have been very severe after the first immunization already, and they were therefore excluded from the booster vaccination. You can see that on the far right that they did not receive a booster vaccination. And I want to conclude with the safety, although again it's not shown in the slide. I want to emphasize that no symptoms suggesting Lyme borreliosis or immunoarthritis were reported throughout the study. Now let's jump to the immunogenicity. The figure here shows the electric mean titers measured by ELISA. On the y-axis, you can see the titers against all of the OspA serotypes combined. So instead of depicting here each single of the 6 OspA serotypes, I decided to put them together and combine them so it's much easier for you to understand the figure. And on the x-axis, you see the different time points with the priming, seropersistence, and booster responses, again, for different dose formulations and dose groups. What becomes quite obvious at first sight is that there has been a substantial increase in antibody titer against all of the 6 OspA serotypes for all doses and for all formulations after the priming. But there hasn't been a marked increase after the booster response, and that booster response is indicated by the red bars. So also after the booster immunization, the use of the adjuvant had a significant positive effect on the antibody induction, as you can see here on the very left side, with a 30 microgram adjuvanted dose inducing the highest titers overall. So based on what I've shown you so far, so the favorable tolerability profile and the high post-booster antibody titers, the 30 microgram adjuvanted as well as the 60 microgram adjuvanted dose were identified as the optimal doses respective formulation. And therefore, those 2 doses have been carried on into the second part of the study. And on the next slide, I just want to show you, I hope it's a bit complicated, the slide. I want to show you just for the 30 microgram dose reverse cumulative distribution. And so that's now each individual serotype, each individual of a serotype, not put together, but you can see the single ones. You can see at different time points the percentage of participants achieving a certain antibody titer. And what becomes clear is that after the priming vaccinations, most participants had received, and the participants that had received, of course, 30 microgram adjuvanted formulations had antibody titers of at least 1,000. with, for instance, 52% achieved titers of at least over 5,000. And after the booster, that even increased to more than 80% had antibody titers of at least 10,000. So we have no idea what those antibody titers mean or, well, how they correlate to protection, but at least we are sure that we generate high titers of the antibodies. And in addition, and again, that's not illustrated here, I just want to mention that further to the high titers of the OspA antibodies measured by ELISA, we could also show that these antibodies are also functional, which means that they are able to bind to the surface of Borrelia and promote the killing of Borrelia species. And again, what I also want to say is that those antibodies, and the functional antibodies, very nicely correlate with ELISA data that I showed you before. And with this, I want to conclude and quickly recapture the presented data on the slide here. So I showed you safety data highlighting the superior tolerability of the adjuvanted 30 and 60 microgram formulations after the priming schedule. What I did not show you, but I want to mention now, is the data from the second part of the study, where the OspA vaccine was equally well tolerated in individuals previously infected with Borrelia burgdorferi sensu-lato. And when summarizing the immunogenicity data, it's important to note that substantial mean GMGs were introduced by all formulations after the primary immunization schedule, that a positive effect of the adjuvantation on antibody titers following the booster immunizations was demonstrated, that a high antibody response induced with the adjuvanted 30 microgram dose after the booster immunization, and keep in mind that has been demonstrated in a seronegative population. And again, from the second part of the study, where the 30 and the 60 microgram formulations were compared, a significant higher antibody response following vaccination with the 60 microgram adjuvanted dose was achieved in the seropositive population, demonstrating a clear dose effect. And lastly, the functional antibody results confirm the anti-OspA IgG ELISA results. So what overall is to say that combining now the totality of the tolerability and immunogenicity results in a seronegative and also the seropositive population, they clearly favor to proceed with the adjuvanted 60 microgram dose into a pivotal phase 3 efficacy study. And with that, I want to end, and thank you for your attention.
Thank you, Dr. Resnick. We really appreciate that, and especially we appreciate you joining us from Europe. So it's much later there today. So thank you for that. And so now we'll move to our summary and conclusions. If we can pull up the slide here. And this will be provided by Dr. Larry Madoff. Dr. Madoff is an infectious disease physician specializing in the epidemiology of emerging pathogens, bacterial pathogenesis, and international health. He is a professor of medicine at the University of Massachusetts Medical School and lecturer on medicine at Harvard Medical School. Dr. Madoff has directed ProMed, the Program for Monitoring Emerging Diseases, since 2002. He is a member of the American Society of Microbiology, the International Society for Infectious Diseases, past president of the U.S. Lancefield Streptococcal Research Society. He is a fellow of the Infectious Diseases Society of America and a fellow of the American College of Physicians. Dr. Madoff is a graduate of Yale College and Tufts Medical School. He performed his internal medicine residency at New York Hospital Cornell Medical Center and his infectious disease fellowship at the Harvard Medical School Longwood program. So, Dr. Madoff, I turn it over to you.
Thank you, Dr. Beard, and thank you to all of the speakers for their excellent presentations. And also I wanted to thank HHS and CDC for organizing this webinar and for all of the participants. We've had a good turnout for this webinar, and I appreciate all of your attention. As editor of ProMed, I see a lot of news of Lyme disease, and I think that one thing that may not have been emphasized enough during this webinar is the tremendously rising rates that we're seeing, at least in the U.S., of Lyme disease. That's attributable to a number of factors, but just yesterday on ProMed we posted a report from Pennsylvania which showed a near doubling of cases of Lyme disease between 2013 and 2014, which was somewhat geographically irregular. In other words, it wasn't the same throughout the state, but this is very relevant as we go forward. I have up here a slide, and I hope the animation works. I'm going to try to do this. So maybe Ben, if you can do this, you can help, but maybe it's not working. This is supposed to be showing the incidence of the number of confirmed cases of Lyme disease from 1988 to 2013 in Massachusetts.
I can't seem to make it work on my end, but maybe you can describe what's going on with it. Sorry about that.
Okay, no problem. I was afraid this wouldn't work, but this is a slide that Dr. LB Maria sent to me. It's and it's from State Health Department data. He's the state epidemiologist from Massachusetts, and it just demonstrates in Massachusetts, which is a relatively high incidence state for Lyme disease, really what's been a steadily rising number of cases from a handful back in the 1980s to thousands of cases that are reported each year at this point. It's also shown the geographic spread of Lyme disease from the Cape and Islands and a little bit of the North Shore of Massachusetts to now Lyme disease being seen throughout the state. And I see this. I now see patients out at University of Massachusetts, which is in central Massachusetts, and as a clinician, we see many cases of Lyme disease, both early and late manifestations occurring throughout the state. And so I think that's the background, and I think part of why Lyme disease, the Lyme-Rix vaccine may have been lost to us is that it occurred at a time when the incidence of Lyme disease was not nearly as high as it is today, and I think now we're hearing much more of a demand for a Lyme disease vaccine than we might have seen then. I'm going to sum up very briefly some of the points made by each of the speakers, and then I will open to questions during what I think will be the last 10 or 15 minutes that we have. First, Dr. Plotkin introduced the topic and reminded us through a very personal anecdote just how close to home Lyme disease has hit many of us, with really a near-death experience of his own son with Lyme disease and tertiary manifestations of Lyme. And also his experience as a vaccine developer and realizing that vaccines are effective prevention, and often our behavioral interventions are less effective in preventing disease. He also underscored the pretty lukewarm recommendations that came with the initial introduction of the Lyme vaccine, and including that it really was not ever a recommended vaccine. It was really just a vaccine that was meant to be considered in certain populations, and that it was never really addressed in children. He went on to talk about some of the newer approaches to vaccines, and Dr. Marconi introduced us to the concept of chimeric protein-based vaccines, and his own really exciting work on chimeric vaccines expressing numerous epitopes. He called them chimeritopic vaccines or Chimeritope vaccines, and introduced us to three of the outer surface proteins of Borrelia, the OspA, which is expressed primarily in the tick, and then Ospc, which is expressed more in the mammalian host, and OspE, a later expressed antigen. And he also introduced us to the heterogeneity in each of these surface proteins, and the need, therefore, for a vaccine to contain multiple epitopes in order to have activity against the vaccine in various phases. As you're looking at CDC data on the spread of Lyme disease, both numerically and geographically over the last 15 years or so, and it shows it just as well as the animation I was trying to show you did. So Dr. Marconi walked us through some of the steps in vaccine development, epitope mapping, showing that antibodies to various epitopes would be bactericidal, and then he showed us how these components could be assembled into a chimeric protein vaccine and used in experimental animals, and also the need in these chimeritopic vaccines to avoid conformational epitopes and use linear epitopes. Dr. Aronowitz really helped us understand, I think, some of the social constructs that surround Lyme disease, including the framing of this disease as a new disease, which perhaps it was not. It was newly described in the U.S., but had probably been occurring in Europe for some time. But the consequences of calling it a new disease and the emergence of an orthodox and heterodox viewpoint on the disease itself, and then how those orthodox and heterodox viewpoints were also a lens through which vaccine was viewed, and also led to conflicting views on the safety and efficacy and the need for the vaccine. And also how the public health case was for use of Lyme vaccine was somewhat limited compared to other diseases which, for example, are contagious from human to human, so there's no evidence of herd immunity in a Lyme disease vaccine. And he emphasized the need for less hubris as we approach these new vaccines. Dr. Resnick introduced us to a Baxter vaccine, which is also a chimeric vaccine and includes two OspA epitopes, a type 1 and a type 2, and showed us how this vaccine was introduced in a clinical trial, a phase 1/2 trial in a reasonable sized population with 300 people in a dose escalating trial comparing adjuvanted and non-adjuvanted vaccine. They used alum as the adjuvant in these trials, which is approved and used in human vaccines, and demonstrated safety in these trials with a lower incidence of adverse events in the adjuvanted vaccine groups, and also showed that these vaccines could elicit antibodies, especially when used with a late booster, and showed it using very nice reverse cumulative distribution plots that there was good response at a population level. However, there obviously is not yet demonstration of efficacy in this vaccine trial. So I'm going to end the summary there and open the forum to questions. There already are some questions that have appeared on the screen here, and at this point you're only able to ask questions using the web interface. And as Dr. Beard instructed, please do identify yourself when you ask the question. So I'm going to start with questions. I'm going to start with a question from Michael Perry. And this question is directed to Dr. Marconi or Dr. Ressing. Do any of the antigenic variations in OspA for the newer vaccine candidates address coverage for Borrelia miyamotoi, a newer strain of Borrelia? And so this is Michael Perry from Connecticut. Dr. Marconi, can I have that question go to you first?
Sure. The answer to this question, Michael, is that the chimerics that we are designing do, in fact, include epitopes that would provide coverage for Borrelia miyamotoi. So that, I think, is one of the attractive features of designing epitope-based vaccines, is that you can essentially incorporate any epitope that you choose, whether it be another Borrelia species or even an epitope from a pathogen that causes a very different disease.
Okay, thank you. The next question is from Heidi Smith. And the question is, I think, to Dr. Ressing, are there any updates on when a phase 3 study of the next generation vaccine from Baxter might begin?
Yes. Well, as you might know, that Baxter has divested their vaccine department to an external company, to Pfizer. And I think that has been going on for since last year. Lyme vaccine has not been sold to Pfizer. Unfortunately, I don't know what the stage is at the moment.
I can comment on that. At the moment, there is no developer for the former Baxter vaccine. However, there are biotech companies that are considering obtaining the vaccine from Baxter and developing it further.
Okay. There's a question from Jill Auerbach, and it's a lengthy question that I'm not going to read the entire question. She's from the Dutchess County Legislative Tick Task Force in New York State, and her comment is that it's past time for the need of a Lyme-only vaccine, and that what is needed is an anti-tick vaccine, like Dr. Whitekel has been working on. If ticks cannot remain attached, they cannot transmit disease pathogens, including Lyme, Babesia, anaplasmosis. So I'm not sure who to address that question to. Maybe Dr. Plott can take that as sort of the long view.
Well, the point is well taken, and I did on one of my slides mention tick proteins as possible components of vaccines. But one has to build on things. The knowledge that we have is that an OspA vaccine will have at least moderate to high efficacy. As Dr. Marconi outlined, additional proteins, additional antigens might well increase the efficacy of that vaccine. The addition of other proteins, including tick proteins, could provide protection against other tick-transmitted diseases. But in vaccine development, one has to build on knowledge to achieve goals one by one. And what we know is we can prevent Lyme disease. We do not know yet for sure whether we can prevent all tick-borne transmitted diseases. The research that needs to be conducted, that will be conducted, will tell us whether that is possible. And if it is possible, then certainly such vaccines will be made.
Thank you. The next question is from Andrew Lima, who asks, for those of us in state and local health departments, how can we improve our communication with the public regarding the past and current state of Lyme disease vaccines? And perhaps I'll send that question to Dr. Aronowitz.
I think something like what we're doing right now is part of that effort, to post information about what we have learned from past experience and exciting new developments. Unfortunately, in this void we're in, especially with the vaccine that has most potential being orphaned, I think it is a time of great uncertainty and anxiety for those of us who would like to see an effective vaccine be developed.
And this is Ben Beard. I'll also chime in on this because I know there was a question maybe about for those who were not able to catch this webinar, the video portion of this. Yes, this will be posted at our website, and it may take a few weeks to get it up there, but the audio and video portion of the entire webinar will be posted, and that is at www.cdc.gov/Lyme.
The next question is from William Nicholson at CDC, and it's for Dr. Marconi. Do the Chimeritope vaccines require carrier proteins or adjuvants to induce protective antibody levels?
The answer to the question is that they do not require carrier proteins. They are very immunogenic on their own, so that is not required. But we have, as a matter of course, consistently pursued using adjuvants, specifically using alum, since it is approved for use in humans. So we have not directly compared adjuvanted with non-adjuvanted Chimeritopes, but we have shown very effectively, I believe, that alum, which is approved for humans, works quite well with these Chimeritopes.
Great. I'm sorry, I missed a question from Sebastian Franco because there was also a question about the availability of the slides and audio that Dr. Beard answered. But he asked, are there any policy recommendations to deal with Lyme disease? And I'm not sure who that question, maybe Dr. Beard actually from CDC might be able to take that.
Yeah, as I mentioned, the webinar will be posted, slides and audio, after the call. In terms of policy recommendations, I'm assuming that this refers to recommendations on a vaccine if and when one were available for use. And Dr. Plotkin can comment on this further, but typically those types of policy recommendations come from ACIP, which is actually a federal advisory committee that is apart from CDC, but it makes recommendations to CDC on the use of vaccines. And I don't know, Dr. Plotkin, if you would like to comment on that.
Well, I'll try not to wax eloquent, but indeed, as Dr. Beard said, it will be the ACIP that will make recommendations. What I detect, fortunately, is a shift in opinion about the importance of Lyme disease prevention. And on the condition that that a vaccine developer does the proper studies and approaches provision of the vaccine and advertising of the vaccine in the manner which I discussed, I think such a vaccine will be recommended and will be used. Now, of course, there are many aspects that go into a recommendation, whether boosters are needed, if so, how often, whether children can be vaccinated, all kinds of things that the ACIP considers. But I am optimistic that given a licensed vaccine, that it will be recommended.
The next question is from Andrew Palm, and I think maybe addressed to me. Do you believe that the increase in Lyme disease incidence in Massachusetts and beyond from 1988 to 2013 and the reported increase in the past year is attributable more to increased public provider awareness and diagnosis or increased prevalence of the disease in ticks? I think yes. I think it's due to several factors, including increased awareness, including increase in the actual rate of disease, and perhaps also some changes in case definition. I think there are important factors that are driving the emergence of Lyme disease include migration, the movement of populations into areas that were previously completely rural and now have increasing amounts of population. I think it's been shown that Lyme disease occurs particularly in areas where there's a lot of forest edge, and the population of Massachusetts has increasingly been encroaching on areas of forest. So I think there's both real and perhaps artifactual reasons for the increases in Lyme disease.
Dr. Madoff, I think at this point we're at the bottom of the hour, or the half hour, and so it's time for us to conclude our webinar. I know there are a couple more questions, and so we'll try to handle those by email back to the persons who submitted those questions. And so with that, I want to thank the speakers and also want to thank all the participants for taking time to listen in on the webinar. And this, as I mentioned, this will all be posted. So with that, we will bring it to an end. Thank you.
Thank you for your participation. That does conclude today's conference. You may disconnect at this time.